In this video, we will discuss about role of carbohydrates in immune cells, blood vessels, and intestine epithelium. In terms of immune cells, we will cover both the innate immune cells and the adaptive immune cells in brief. So, whenever a pathogen that has the carbohydrate structures enters the host, it can be recognized by the pathogen receptors that are present on the innate immune cells. And the example here is shown of the bacteria. So bacteria has the glycocalyx layer that covers the membrane of the bacteria. This glycocalyx layer is made up of carbohydrate structures or the polysaccharides, and those polysaccharides for the host cells are known as this PAM, that is pathogen associated molecular patterns. And these PAMs can be recognized by the receptors that are present on the innate immune cells. Those receptors are known as PPR. And two of those examples are shown here, that is toll-like receptors and the mannose binding ligand. So when the these PAMs interact with this TLR, it can trigger intracellular signaling pathways that will leads to the upregulation of the inflammatory genes, and that will trigger the immune response or inflammation response against the pathogens or the bacteria in this case. In the other example, when the bacteria or the glycocalyx on the bacteria interact with this mannose binding ligand, it can trigger the complement cascade. It will trigger the cytolysis of these pathogens in the extracellular matrix itself. So this is about the innate immune response, where carbohydrates plays a role towards immune response against the harmful pathogens. Now we will understand the adaptive immune cells, that is T cells. So in this case, antigen, the bacteria will enter the T cells, and thereafter this PSA depolymerization occurs with the help of this reactive oxygen species, and this depolymerized PSA depolymerized can interact with this major histocompatibility complex, and after that this major histocompatibility complex can provide those information to the T cells. So thereafter T cells can get activated and can trigger the specific immune response against those bacteria or against the antigens for which this information is being provided by the major histocompatibility complex. So one thing I need to correct myself is that here the bacteria is not entering into the T cells, however it is entering into the normal cells, from where the major histocompatibility complex provide information to the T cells. This is about the immune cell response directly. Now we will understand how the carbohydrates plays a role in the case of these blood vessels. So blood vessels contains various selectins at its endothelium surface, such as E-selectin, P-selectins that can interact with this ligand, that is PSGL1 ligands that are present on the leukocytes that are circulating within the blood flow. So whenever there is a requirement of any requirement of the leukocytes at any site of infections, this expression of E-selectins and P-selectins will be increased, and that will increase the chance of these leukocytes to get adhere to the E-selectins and P-selectin. Thereby it will cause leukocyte adhesion, and thereafter it will help in the intravascular of these leukocytes to reach at the site of the infection. So this is in brief about the blood vessel endothelium, role of carbohydrates in blood vessel endothelium. I have prepared a detailed video. You can go through that video to understand about this topic. Lastly, in the end, we will discuss about this intestine epithelium. So in this case, the glycocalyx layer on the bacteria can also interact with these glycans that are present on the epithelium of the intestine. So in this case, and it can leads to different function. For example, the initial phase that begins with this adhesion of this bacteria's glycocalyx to the receptors that are present on the intestinal epithelium. And because of this, what will happen is that as soon as adhesion occurs, it will lead to the triggering or it will trigger the virulence, uh, virulence upregulations of these virulence genes, and because of this the virulence genes will lead degrade himself. The upregulation of the virulence genes will lead to the production of this toxin that is harmful to the bacteria, and thereby this can act as a feedback loop where as soon as the bacteria detects the receptors on the intestine epithelium, it can trigger the immune response in terms of these virulence genes. And another thing that can occur in this case is that most of the bacteria has the ability to manipulate this to manipulate or utilize these ligands in a way that they can degrade those uh, ligand, and at the same time they can modify the Glycan structures that are formed on the intestinal epithelium, such as the more fucus-related Glycan structures are formed on the intestinal epithelial cells. Thereby, they can control, in this case, the bacteria can control both the virulence genes and also the bacteria productions within the intestine, which is harmful to the human being. So I hope you got a basic idea how this carbohydrate, what is the roles of the carbohydrates in the immune cells, blood vessels, and the intestine epithelium. If you need to understand more about this, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.